development is in an ever-changing situation. But over the past 10 to 15 years, this term, new approach methodologies, has really come into the spotlight. And today I'll be talking to you about whether or not they can actually fully replace animal testing. So as I mentioned, we're in this new era of drug development where regulators, such as the US Food and Administration, also known as the FDA, have recently come out and said that they'll support the use of non-animal methods in order to bring a new drug to market. The UK government, in November 2025, also released a roadmap to ending the use of animals in drug development. And the use of animals has been around for nearly 100 years. You've been legally required to test on animals in order to bring any drug to market. Now, these changes, no one is ever 100% comfortable with change, but they have really divided the scientific community. So what does this roadmap that the UK government released really look like? Well, to put it in perspective, almost every drug that you will have ever heard of has been through some sort of animal testing. From your first-line chemotherapy treatments, such as doxorubicin and methotrexate, but also your everyday headache cure, paracetamol. Also, compounds that have been awarded the Nobel Peace Prize have been through animal testing, such as nivolumab, which won the Medicine and Physiology Prize in 2017 and is hailed as one of the breakthrough immunotherapies for lung cancer. In the UK alone, in 2024, 2.64 million animals were used in scientific procedures, almost a quarter of which were used to knock down genes to understand how diseases can occur, something that new approach methodologies is very close to being able to replace by taking those cells straight from patients. Regulatory and ethical challenges also arise when we talk about ending the use of animals. As I said, nearly 100 years has passed since we decided that we had to use animals to bring any drug to market. That means there's a lot of tightly interwoven laws and regulations that need to be unraveled and rewritten. But is this roadmap a little bit too ambitious? For example, an area of drug development that heavily relies on animals in the current situation, pharmacokinetics, really is sort of penciled to be the first one to replace animals. And by 2030, the government wants to reduce this by 35%. And this is an area where new approach methodologies aren't really ready to do anything in. Like I mentioned, some areas are closer than others to replacing animals. And I'd like to say that given another 10, 15 years, the majority of these will be replaceable. But the issue is not developing these new approach methodologies, it's implementing them on a wide scale. It's all well and good saying that this company can have this new approach methodology, this university is working on this one, but in order to get drugs to market, there needs to be a uniform blanket ruling. For example, most new drugs go through what's called a cell killing assay, and that relies on an assay that's been around for 45 or so years now called MTT. MTT is widely proven to be superseded by other cytotoxicity or cell-killing assays, such as the LDH assay, but MTT is so widespread across different countries, etc., for drug development that it's still written into regulations. That's not to say that the MTT assay doesn't work. As you can imagine, writing any new laws or changing regulations takes a lot of time. But the biggest thing that will take the most time is changing our habits. We've relied on animal testing for so long. How can we go about not relying on them further? This roadmap or this movement of new approach methodologies has created a bit of a scientific divide. And imagine this like your classic bell curve, where the majority of people sit in this middle section. And the majority of these people believe that one day new approach methodologies, whether it's in 10 years' time or 50 years' time, could fully replace animal testing. But in order to do that, there's requirements that need to be set. And that is careful testing to make sure that they actually replicate what happens in the human body. And also validation of these. So exploring and unturning every rock so that we don't have any clinical failures and disasters that create, patient death, for example. On one end of this bell curve, you have people that are incredibly against replacement of animals. And this is partly because they recognize the positive and success of animal testing that's gone on for, like I said, nearly 100 years now. And there's no doubt that animal testing has been beneficial for drug development. We've had some fantastic drugs, like nivolumab, the Nobel Peace Prize winning compound. But just because we've always done something doesn't mean that we should continue to always doing it. And if we have these models that are more predictive of what will happen in human biology because we're using human cells rather than animal cells, why shouldn't we move with the times and keep on moving forward with progress? And the other reason why people in this sort of against animal testing are sort of in this camp, let's say, is because they don't want their scientific achievements of their long-term career to be sort of dismantled and disregarded. And I really don't think that would happen, because just because they've used animals doesn't mean that they are wrong to have done that. They've used the best technology they have available. On the other side of this bell curve are the people that believe new approach methodologies should happen like that. And this is because they believe that this gap that's created by not being able to use animals will suddenly see a huge investment from pharmaceutical companies and the development of new approach methodologies. But we run the risk of not being able to achieve this straight away, and running the risk of then not bringing new therapies for patients who need it for diseases like cancer. So, what actually are new approach methodologies? This figure, taken from a review from last year, really sort of sums it up, with the top being the traditional method of animals. And there's a governing body that regulates animals under 3 R's: replacement, reduction, and refinement. And new approach methodologies look to throw the animal testing out of the window through 3 systems that combine together to give us better human predictivity. The first is simplified organoid structures, where we can grow simple, for example, cancer or liver tissue in a lab that just has that particular disease tissue in. These give some really nice morphological and relevant information, but they lack the ability to flow compounds through a blood network. We then have the more sophisticated older brother, if you like, organ on a chip. Now, these are able to have multi-cell structures, which is replicative of organs, because one organ is not made up of one type of cell. You're also able to flow these drugs through a vessel network, and there's some lovely slides that you'll see in a couple of minutes. And this allows us to then chain different organs together and start to replicate the human body in a laboratory. And the other area, which you're probably not surprised about because it's in every area of sort of tech at the moment, is AI learning. And this machine learning enables us to rapidly screen thousands of drug compounds at a really quick rate to see and predict whether they will cause any damage before they go into our patients. Combining these together will put more confidence in our predictivity of what will happen in humans, and also put pressure on regulators to move with these times and enable this for drug development. And new approach methodologies not only look to replace animals, but they also have this new area of personalized medicine. Imagine I have lung cancer. Imagine the ability to go to the doctor, get a biopsy, and send those cells off to the lab, and then grow my tumour in the lab with blood vessels and screen what drug therapies will work best for me. That enables me to get the right treatment that's going to work for me. And in cancer, that's so important because a lot of chemotherapies will cause serious side effects to most patients or resistance. So by being able to streamline and use the one that's most appropriate for me, removes that off-target toxicity effect for me and improves my quality of life. And just to summarize this section, this graph is one that I've used for many years, and it really highlights the traditional way using animals in reaching that sort of human relevance that enables us to make that decision to move into clinical trials. And new approach methodologies get us there much faster. And this enables multiple things, one of which is saving time and money for developers. You may not be aware, but it costs up to $2.5 billion on average to bring one new drug to market. And that takes approximately 12 to 15 years. So that's a lot of time and money if we get a drug 14 years down development and $2 billion to find out that it now no longer is suitable. So if we can eliminate them much sooner or progress the lead compound with greater confidence, this will enable more streamlined drug development processes. And an example is this liver on a chip model developed by a well-known organ on a chip company. And the liver is a really important organ for assessing toxicity of drugs. If you have, I'll use the example of lung cancer again, and you're given lung cancer, the main organ that this drug will be released in, other than the lung cancer, is most likely your liver, because it breaks down pretty much anything that your body takes in. So using the liver to predict toxicity is really important, but this group went even further by showing that they were able to predict clinical outcomes or human patient outcomes much better than using animals. And they suggest that this model actually be implemented just before animal testing in drug development. And this means that we can rely on enhanced confidence, but also eliminate those drugs that originally would have been taken through for lead. So just for a bit of background, with this method, you may take forward 2 lead compounds for animal testing. And you may be thinking, oh, well, that's still using animals. But without that, you may have to take 5 or 6. So you're significantly reducing the amount of animals. And eventually, these methods will get more advanced where we can say, actually, we may not need any animals. So it's a replacement through reduction method. Now, this is a slide I was talking about earlier. And that lung cancer example that I gave, where you take my cells, this is exactly what happened for these patients that had breast cancer. So these are cells that have been taken from a patient's tumour, taken to the lab and grown up, and I've zoomed in on patient 2, and that white centre is the tumour itself. And this orange sort of woven web is actually blood vessels. And what they did was they treated these cells using drugs that they treated, going through that blood vessel and into the tumour to show an anti-cancer effect, i.e. treated this cancer, which really highlights the advance in technology. Sort of 10 years ago, no one ever thought that this may be possible. And it's impressive to see how quickly this has come about and how relevant this is to our ever-growing drug development crisis. And shifting gears now to a company that is actually a 3D bioprinter. They have successfully managed through this research group to print neuronal network cells. And if anybody's ever seen neuronal networks, this is exactly what they look like in our brain. These sorts of long stranded branched networks. And you can see here, the bottom right, these 2 images. One is looking from a bird's eye view and the other one is looking from the side. And this proves that this network is actually 3D. And this is really important for studying diseases like Alzheimer's, Parkinson's disease, which has seen very little advancement in therapeutic approaches for the last 20, 30 years. And these are so sophisticated that actually I was at a demo yesterday by this company, and they printed an ear to see, which is, I think, fascinating. So I've talked a lot about new approach methodologies and how they could replace, and I think the real question is not, can they replace? I think that day will come. It's how soon will that day come? And how soon relies on this simple equation of an openness to use and adopt these new approach methodologies and really start to push the boundaries of how far these can go. Followed by the changes in this legislation and untangling that tight mesh of laws and regulatory sort of standpoints, but also to make improved ethical decisions. Nobody wants to use a defenseless animal if they could use an alternative method, i.e. taking some patient cells, which causes no damage to the patient or very little, and to inform more relevant human systems. And through that, we will get a reduction of animals and eventually the full replacement. Thank you very much.